Hi everybody, welcome back. So in our last lecture, we ended with sort of the advent of penicillin and understanding that there are differences in prokaryotic bacteria with gram-negative and gram-positive types. So now in this lecture, we're going to start to look specifically at why that is and what specific features um, start to help us to be able to understand these differences. So the first thing I want to talk about is ribosomes. Oops, sorry. So ribosomes are responsible for the creation of protein in both prokaryotic and eukaryotic cells. However, when we're looking at a prokaryotic ribosomes, there are specific antibodies that can actually disrupt this ribosome function. So rather than only targeting these cell walls, there's potential for antibiotics to disrupt uh, things within the cell. And because it has this specific folding, no protein is formed, and the antibiotics interfere with protein synthesis, so the cells aren't able to continue. However, when applied to eukaryotic cells, it has a slightly different function here, and it's not able to bind and disrupt the ribosome and the protein function, so you still are able to get those nice protein folds that we talked about in our previous lecture. So that is one of these. This is looking specifically at the structural differences that can occur between prokaryotic and eukaryotic cells and why an antibiotic is able to disrupt specifically prokaryotic cells and leave those eukaryotic cells to be healthy and continue to create functional proteins. Another big main difference that is really important for the development of different antibiotics and different ways that you can attack these cells is looking at these cell membranes. So a cell membrane, as we talked previously, are made of these phospholipid bilayers. So this is a lipid here, this phosphate lipid, where it has those hydrophilic phosphates on the outside, and then these hydrophobic strands on the inside of the cell. And it forms this bilayer. And what's important about this is that it is semi-permeable. So some liquid is able to pass through the cell membrane. It's not completely encased or relies on specific active transport. There's some passive diffusion that can happen across this bilayer, and that ultimately is what helps these cells keep its structure. So what I mean by diffusion is specifically osmosis. So osmosis is the diffusion of water across a semi-permeable membrane. And what's important about this is understanding the direction that osmosis occurs. So osmosis always occurs across a semi-permeable membrane from an area of low solute concentration to an area of high solute concentration. So what does that mean specifically? So for example, in a hypotonic solution, we have this higher solute concentration inside the cell. And that means that the water is going to flow into that cell, and the cell is going to expand. The opposite of that is a hypertonic solution, where you have a higher solute concentration outside of the wall, and you have then water flowing outside of it, and this cell starts to shrivel. And then in this central state, we have basically a state of equilibrium, where the solute concentration is the same inside and out, and water flows equally in each direction. A good example of sort of thinking through this is the reason why you can't have an IV of only water, because if you have have something of only water, you have you can create these hypotonic solutions where essentially water is going to start flowing into these cells and your cells are going to swell. Um, so it's always important to have some salt content in there so you can have essentially an isotonic solution when you're receiving fluids. And this osmosis is able to occur because we have this again phospholipid bilayer, and that is both hydrophobic and hydrophilic. So because it's able to create this barrier and have this this structure, you're able to maintain a fluid both on the outside and on the inside of the membrane. And importantly, not only does it sort of allow for some passive diffusion, it also is like it prevents many large molecules such as glucose and hydrophilic charged substances like sodium ions from crossing, while also allowing small uncharged substances to cross just via diffusion, just naturally being able to flow through this phospholipid bilayer. So simple diffusion occurs when you have sort of this natural tendency of dissolved substances to move from one area of high concentration to an area of low concentration. And what's important about passive diffusion or simple diffusion is there's no energy required by the cell for this to maintain. And again, this sort of keeps both the structure of the cell constant when we're thinking about the overall structure of the cell. However, how does that work when we have sort of these larger molecules? How do they cross the membrane? Well, we have specific passages that are transport proteins, which are proteins that span that phospholipid bilayer and can assist in diffusion of larger molecules. So in facilitated diffusion, we have again this transport protein, and large or hydrophilic solutes can move across this membrane from, again, going from higher concentrations to lower concentrations and allow these larger molecules to pass through that wouldn't be able to pass through simple diffusion. And this does not require any sort of energy from the cell. Then we have something much more active. We have active transport, where solutes are literally pumped from one area of lower concentration to higher concentration with the help of transport proteins. So now we're getting sort of the opposite. This requires energy. We're taking molecules from inside the cell and passing them out. And this requires energy from the cell. So we call this active transport. So how do these proton pumps relate to our questions of antibiotics and how they target prokaryotic cells? Well, antibiotics can actually cross those prokaryotic cell membranes using those transport proteins. So they're able to gain access to the cell because they're able to fit through those active transports. And they can also, but likewise, bacteria can start to resist antibiotics using transport proteins. So there's a little bit of a give and take there that can occur between these mechanisms. So you can quite literally pump out the antibiotic. Okay, so I think that sort of concludes thinking through some of the structural elements that really are important for different antibiotics and the overall structure of the cells. So we're going to pick up next time where we're going to look more specifically at the organelles and the large structures that are involved in the structure of cells. Okay, see you soon.